Hello again. Thanks for joining me. Let's pick up from where we left last time. So we were looking at this slide and remember the terms genotype and phenotype. Genotype was what is coded in the genes, and phenotype is what we can see as an output. And we also talked about the fact that there are two alleles for each characteristic. These alleles are our genotype. And what we see, like this man here, is our phenotype. So let's take this information and apply it as Mendel did. And there is a lot of stuff going on in this slide. Do not let that overwhelm you. We will go through it all in steps. So what we see here is really just terminology, nothing to be worried about. So as we are studying different generations of plants in order to follow how certain traits are passed on from parents to offspring, we need to be able to identify that what was generation we are talking about. And as I am sure that you have figured out by now, we scientists love shorthand. So what we do is that we call parents as the P generation. And then the first round of kids, we call them TF1 generation. But here comes the beauty of Mendelian genetics. We go further than just one generation and their offspring. We look at the babies of the first round of babies. So this would be our second generation of offspring called F2 generation. So that is really all there is in terminology with that. And the term gametes, you have already heard before. They were the sex cells, either eggs or sperm, or in case of plants, either the equivalent to egg or sperm. Let's see what else can we see in this diagram. Yes, we saw that the parent generation produced sex cells, gametes. And we see that for those sex cells, we see that some are marked with a capital letter and others are marked with a lowercase letter. How strange! Well, these letters are symbols for our genes. So the genetic coding for a certain characteristic. Say some garden peas are green and some are yellow, right? So in this case, we know that we typically see more green garden pea pods than we do see yellow ones in these plants. But that does not mean that yellow ones would not be an option as well. The reason why we see more green garden pea pods is that it is a dominant characteristic. And we mark that with a capital letter. And since the recessive characteristic is less likely, we mark it with a lowercase letter. So what does that all mean? Why does this matter? Well, let us go back to this slide just briefly. Remember, we had two alleles for a single trait, right? What we are seeing is that sometimes some of them are the exact same, or they might differ. Well, what happens is that you have a characteristic that is dominant. In this case, it would be the pea pod being green in color. It is what we see. And we will see if that both of these alleles were dominant, or even when just one of these alleles is dominant. It always takes over if it's present. The only time that we will see regressive characteristic taking place in the appearance, remember we called this as a phenotype, is when we have no dominant one to override it. So we need to have recessive allele for both of these letters. So we must have inherited a recessive characteristic from both parents. remember this diagram. Here we would see a dominant characteristic. And here too. And this would be the only time that we see yellow pea pods, as they are a recessive characteristic. We need both two alleles to be recessive for that to happen. Well, this takes us really far with this diagram that we saw before. So let's read what we can tell from it. Well, there are two parents, and this was our P generation. And other one of the parents looks green, so this is simply that its phenotype is green. Remember, phenotype is what we can see as an end result. And the other one has yellow phenotype. For these yellow pea pods, we must have a genotype that is lowercase g and lowercase g, because it was the only time that we saw yellow pea pods if both alleles were recessive. However, if we see a green pea pod, it might be either capital G and capital G for genotype, or it could be capital G and lowercase g. In this case, we are told that the genotype is capital G and capital G. So you remember our process of meiosis. This was the production of sex cells. And we remember that to make a sex cell, we must divide the full amount of genetic material in half, as each parent contributes half of the genetic content. So in this case, the sex cells that we have the parent generation to produce are capital G for green pod and lowercase g for yellow pod. So the question now is that what kind of babies are they going to have? Well, we remember that if there is even just one capital G present, it will override any other form of that allele. So we will see green pods as a phenotype. But if we see that all of the offspring in F1 generation looks green, so have a green phenotype, does this mean that all yellow pods have disappeared? No, it does not. The cool thing here is that you will notice that we have still the lowercase g there. So one of the alleles of the offspring carries a chance for a yellow pod in a genetic level. But we just cannot see it. The phenotype does not show it. So what if we now take these babies that are our F1 generation and have them to have babies? So we will now focus on this lower part of this diagram. And please do not get overwhelmed by it. We will go through everything in steps. Before we study this part in more detail, let's just note that all of the babies in F1 generation were of a genotype capital G and lowercase g. So that is what their alleles are. But their phenotype was always green. So now we take two plants like this and fertilize them together. What kind of babies can we have in the F2 generation? Let's enlarge this part a bit. And we have this beautiful tool to figure out what type of babies we will have. It is basically just this table, 2x2 boxes, with some stuff outside it and some inside it. And this tool is called a Punnett Square. What it does is that it lists us all the possible combinations of babies that may come out of a certain combination of parents. And remember, this is only for one trait, the color of the pea pods in this case. So we take the parent's genotype from here and put it to the outside of this box. In this part, the egg cells, although really in plants they are not called it, but just follow with me nonetheless, and the sperm cells on the other side. Now all you have to do is to fill in the table. The capital G from here goes here, and the capital G from here goes here. So this is the first situation. These parent gametes will give us an offspring with capital G and capital G. Well, both parents were passing an allele of a capital G, so it really is not a huge surprise. And we keep doing this for all boxes and filling them in. This gives us a table like this. And if you are confused now with this work, filling a Punnett Square, I have a video where we will do that step by step coming up. But this is the box that we end up with. Wow. What does it tell us then? Well, we see that there 3 of the babies in F2 generation are going to have green phenotype. Remember, this was that they will look green. And one will look yellow, so it has a yellow phenotype. So the ratio for phenotypes will be 3:1. How about genotypes? Well, here we have the key to genotype. And we get a different ratio of 1 capital G and capital G. 2 capital G and lowercase g, and 1 lowercase g and lowercase g. So phenotypes and genotypes differ in their ratios. I hope that this makes sense. You really have covered quite a lot by now. Again, I will have a video coming up where we will draw all this out. And this is the key part of this chapter that you have already covered. And this slide just tells you that we can get very different outcomes by crossing different kinds of parents. We could draw all these out. And all this knowledge gave rise to what we call laws of Mendelian genetics. And basically this just summarizes what we have just talked about. You can review this with your textbook too. Well, it does not have to be only one trait what we are looking at. It can be also 2 traits that we study at the same time. Say, in this example, the color of pea pods and how tall the plant is. Since we are talking about 2 traits at the same time, we call this a 2-trait inheritance. So we do it as before, but this time we include 2 different genes that we study at the same time. And this gives us twice as big of a Punnett square. And we can go even bigger by studying many different traits at the same time. So this was just 1 trait. And now we have 2 traits, as in our last example. What if we do have 3 traits? You will notice that pretty fast it gets really messy. Now that is a huge table to fill in, and with just 3 traits that we study. See, that is because we have to list all possible combinations. And we get the probabilities of all possible combinations by analyzing these tables. What I'm going to do is to include a link to this Punnett Square calculator into your optional resources. I sometimes find it fun to try to figure it out. But it does not have to be just pea plants that we are looking at. Much of current researchers use fruit flies as model organism rather than pea plants. But the method is the same. We can be looking at, for example, the wing length and body color as our two traits, and we just do Punnett squares as before and get our answers. And actually what is really cool is that all applies to many characteristics that we see in humans. Two examples are whether the earlobe is attached or unattached, or if one has a straight hairline, or what we call as a widow's peak. There are many other characteristics such as whether one has freckles or not, skin tone, hair color, and so on. And really what we find out is that this all applies to what we have just learned about meiosis. So we are going to wrap up here for just now, and next I have some review of how to actually draw a Punnett square yourself. And then we will move on to look at how this all applies to humans. If this has felt like a lot to take in so far, do not worry. This is probably as hard as it will get. Pretty much everything else is just application of these concepts. You have done really well. It is time for you to have a break, and we will see again in a bit.